Hello. Very good afternoon to the student who have joined. Can you please let me know if my voice is audible to you? Let me know if my voice is audible and if you can see the presentation properly. Okay, thanks for confirming. We will start at 4 p.m. Please wait till then. Okay, so it's 4 p.m. I think I will start with my introduction. So, very good afternoon to all. My name is Mousumi Deb. I am a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL course of Medicinal Chemistry. Today, it is our week 12 live session. So, this medicinal chemistry course we have started in the at the end of the January, and we have completed almost the most of our courses. We have completed 11 live sessions till now, and today it is our week 12 live session. This will be our last live session, which is the regular live session, and we will have one more live session on the day after tomorrow. That means on the 13th of April, Sunday from 11 a.m. We will have a summary live session. Okay, so today it is the week 12 live session and of this medicinal chemistry course, and I am going to discuss regarding the various topics of the week 12 course content. Okay, and in the last class, which is on the Sunday, that in that class, what I will do is I will discuss the complete course in a whole. Okay, that day we will discuss some MCQ questions, and the questions will be given randomly, so that I will just want to check that you remember the old things or not. And after that, I will summarize few topics, few points, which I think will be helpful for you for your final examinations. Okay. So before starting the session, like always, I would like to point out few things. First, there will be this kind of problem solving sessions every week, and these sessions will be on every Friday from 4 to 6 p.m. Okay, these sessions are live on Google Meet platform. This is the link for our every Friday live session, and this link will be same, will remain same for the summary session also. It will be notified to you by email. So the most important thing is, like, if any network disruption happens from my side, do not leave the session immediately. I will try to join as soon as possible. I am recording the session. That is why I have muted your microphones and cameras so that I can record the session seamlessly. But the chat box is always open, and I am monitoring the chat box every time. So if you have any doubt, any query, if you want to tell me anything, you just let me know through the chat box. Okay, and the chat box will also you will use to answer the questions which will be asked during the class. Okay, so the chat box will be the medium of our communication. So if you have any doubt, any problem, or anything, just let me know through the chat box. Okay, since I'm recording the session, if there is any network issue from your side, you can just leave and rejoin at any time, because I know you can have the network issues also, but when I will share the complete recording, you can just follow the things whatever you have missed, so you can check it from there. This presentation and class recording, it will be shared to you after the session, and where to find these things I have already mentioned you a couple of times. Again, I will try to just mention you for one more time. Just you have to first go to the NPTEL portal, the medicinal chemistry portal on the SWAYAM portal. This medicinal chemistry course, the announcement section, there will be a left panel where the weekly assignments are given. Here you will find there is a problem solving session January 2025. If you click on that, you will go to this problem solving session recording. On clicking on that particular problem solving session recording, what you will be redirected to a page. If you click on this page, you will find every live sessions, presentation, and recording are in that particular page. Okay, so you can just download the presentations, and you can also check out the previous class recordings when you are going to just revise the things. Okay, so there you will find all the things required, and during the class you can ask your queries or the doubts, and after the class also you can just drop an email to me. I will share my email ID at the end of the session, so if you have any doubt, any query, you can just let me know through my email also, which I will share at the end of the session. So today it is our week 12 live session, and the 12th week course content is the last of your medicinal chemistry course. Till now we have learned different aspects of the medicinal chemistry and drug discovery, and now we have followed all the things, like we have discussed about many therapeutics like antibacterial, antiviral, anti-cancer, and all those things. We have learned what are the principles of medicinal chemistry, what is the pharmacokinetics, pharmacodynamics, and all the other aspects of medicinal chemistry. Most of the thing we have already covered. So what are the course content of today's? So today mostly we will discuss on the QSAR. So QSAR and SAR. SAR we have already discussed, like structure activity relationship, like how the structure of a particular drug, how various functional groups they attribute their function, their biological property. Okay, like we have a compound, it has some certain functional groups, how that functional groups or how the structure of that particular compound is contributing to its biological activity, that is known as a structure activity relationship. One more thing is there, which is known as the quantitative structure activity relationship, and here we use different various mathematical equations and all those things to ascertain this structure activity relationship. Okay, so mostly we will discuss today is the QSAR, and also we will discuss regarding various drug resistance strategies, how what are the various methodologies, or what are the various pathways through the drug resistance happen, and also we will discuss a little bit about the synergy. Okay, so that is the week 12 of our course content of this week. So let us start with the first question of today's session. So the problem number 1, it says that which of the following statements is false regarding the QSAR? Your options are: number A, for the QSAR study, the compound studied need not to be structurally related. Number B, correct testing procedures must be used. Number C, in vitro tests carried out on an isolated enzymes are relevant. And number D, none of the above. Which of the following statements is false regarding the QSAR? Number A, for QSAR study, the compound studies need not to be structurally related. Number B, correct testing procedures must be followed. Number C, in vitro tests carried out on isolated enzymes are relevant. And number D, none of the above. You have to identify the statement which is incorrect regarding the QSAR. I will wait for some time, then I will check the chat box. Let me know A, B, C, or D. Which of the following you think is the correct answer? Which of the following statements is false regarding the QSAR? Let me check the chat box. Okay, A is the given answer. Let me check if A is the correct answer or not. Okay, so the correct answer will be yes, number A, which says that for the QSR study, the compound studies need not to be structurally related. So basically, the QSAR approach attempts to identify and quantify the physicochemical properties of a drug and to see whether any of these properties has an effect on the drug's biological activity. If such a relationship holds true, an equation can be drawn up which quantifies the relationship and allows the medicinal chemist to say with some confidence that the property or properties has an important role in the pharmacokinetics or the mechanism of action of the drug. It also allows the medicinal chemist some level of prediction. By quantifying physicochemical properties, it should be possible to calculate in advance what the biological activity of the novel analog might be. There are two advantages of this. Firstly, it allows the medicinal chemist to target efforts on the analogs which should have improved activity and thus cut down on the number of analogs that have to be made. Secondly, if an analog is discovered which does not fit the equation, it implies that some other feature is important and provides a lead for the further development. So what are these physicochemical properties or physicochemical features that we have mentioned? Especially they could be any structural, physical or chemical property of the drug. Clearly any drug will have a large number of such properties and it would be a Herculean task to quantify and relate them all to the biological activity at the same time. A simple, more practical approach is to consider one or two physicochemical properties of the drug and to vary these while attempting to keep other properties constant. This is not as simple as it sounds, as it is not always possible to vary one property without affecting another. Nevertheless, there have been numerous examples where the approach has worked. It is important that the QSAR method is used properly and in relevant situations. Firstly, the compound studies must be structurally related. So these things I would like to just highlight for your understanding. So firstly, the compound studied, they must be structurally related. Okay, so that is why our first statement number 1 is false, because it says that the compound studies need not to be structurally related, but all the structures called the compounds need to be structurally related and act at the same target and have same mechanism of action. These things are very important. Secondly, it is crucial that the correct testing procedure is used. In vitro tests carried out isolated enzymes are relevant for the QSR study, as the activities measured for the different inhibitors are related directly to how each compound builds the active site. In vivo tests carried out to measure the physiochemical properties of the enzyme inhibitors are not valid. Okay, this is important, that in vivo tests carried out to measure the physiological effects of the enzyme inhibitors are not valid. However, as both pharmacokinetic and pharmacodynamic factors come to play, this makes it impossible to derive a sensible QSAR equation. Okay. So these are the things, these are the points which are very relevant regarding our QSAR strategies, and these points I would like for you to remember, okay, because it will be easier to you to understand what is the QSAR is. Okay. So if there is any doubt, any query, please let me know. Okay, I hope it is clear. Okay, now I would like to go to the next question. Okay. So problem number 2, it says that which of the following is not a QSAR parameter? Which of the following is not a QSAR parameter? Your options are number A, hydrophobicity, number B, hydrophilicity, number C, steric, and number D, electronic. So which of the following is not a QSAR parameter? Hydrophobicity, hydrophilicity, steric, or electronic? Which of the following is not a QSAR parameter? Hydrophobicity, hydrophilicity, steric or electronic? Let me know in the chat box, A, B, C or D, which of the following you think is the correct answer. Let me check the chat box. Okay, so I got one answer which is saying that it is the number B, which is the correct answer. Now let me check. So for problem number 2, the correct answer will be, yes, it is number B. Hydrophilicity is not a QSAR parameter. So what is QSAR? A quantitative structure activity relationship is a mathematical relationship which correlates the measurable or calculable molecular properties to some specific biological activity in terms of an equation. QSAR attempts to find the consistent relationship between the biological activity and the molecular properties so that these rules can be used to evaluate the activity of new compounds. Understanding QSAR regarding the electronic effects and steric effects and lipophilicity. So physicochemical parameters, various parameters are used in the QSAR studies are the hydrophobicity, partition coefficient, pi constitution constant, steric parameter, Stubbs equation, the Stubbs constant, Verloop steric parameter, electronic parameter, Hammett constant and dipole moment. So these are the physicochemical parameters which are used in the QSAR studies. Okay. So many physical, structural and chemical properties have been studied by QSAR approach, but the most common are the hydrophobic, electronic and steric properties. That is, because it is possible to quantify these effects. Hydrophobic properties can be easily quantified for complete molecules or for individual substituents. However, it is more difficult to quantify electronic and steric properties for complete molecules, and this is the only really feasible for the individual substituents. Consequently, QSAR studies on the variety of total different structures are relatively rare and are limited to the studies of the on hydrophobicity. It is more common to find QSAR studies being carried out on compounds of the same general structure where the substituent on the aromatic rings or accessible functional groups are varied. The QSAR study then considers how hydrophobic, electronic or steric property of the substituent affect the biological activity. The three most studied physicochemical properties are now considered in some detail. So we will discuss it in detail later. So remember, these three are the important physicochemical properties. These are hydrophobicity, steric parameter, electronic parameter. Okay. So now I will go to the next problem. Problem number 3 says, which of the following statements is true? Your options are: number A, hydrophilic compounds have a high value of P. Number B, hydrophobic compounds have a high value of P. Number C, acidic compounds have a high value of P. Number D, basic compounds have a high value of P. Which of the following statements is true? Again, I will repeat. Number A, hydrophilic compounds have a high value of P. Number B, hydrophobic compounds have the high value of P. Number C, acidic compounds have the high value of P. Number D, basic compounds have the high value of P. Which of the following statements is true? Let me know, A, B, C or D, which of the following you think is the correct answer. So which of the following statements is true? Let me check the chat box now. Okay, number C, I have got the answer. Let me check if number C is the correct answer or not. So the statement which is true regarding the value of the P will be, it is number B, which says that hydrophobic compounds have the high value of P. So what is the P? Let us first understand that. So P is the partition coefficient. The hydrophobic character of a drug can be measured experimentally by testing the drug's relative distribution in an N-octanol and water mixture. Hydrophobic molecules will prefer to dissolve in the N-octanol layer of the two-phase system, whereas the hydrophilic molecules will prefer the aqueous layer. The relative distribution is known as the partition coefficient P and is obtained from the following equation: P equal to concentration of the drug in octanol divided by concentration of the drug in the aqueous solution. Hydrophobic compounds have a high P value, whereas the hydrophilic compounds have the low P value. Okay. So partition coefficient or the distribution coefficient is the ratio of the concentrations of the compound in a mixture of two immiscible phases of the equilibrium. The most common physicochemical descriptor is the molecule's partition coefficient is an octanol water system. After administration of a drug will go through the series of the partitioning steps, leaving the aqueous extracellular fluids, passing through the lipid membrane, entering other aqueous environment before reaching the receptor. And in this sense, the drug is undergoing the same partitioning phenomenon that happens to any chemical or separatory funnel containing water and non-polar solvents such as the hexane, chloroform or ether. So this is called a separating funnel. Okay, so here basically this separation of the aqueous and non-aqueous layer actually happens. The partition coefficient or distribution coefficient defined as a ratio of concentration of compounds through mixture of two immiscible phases of equilibrium, a biphase or liquid system phases, especially the unionized solutes, and the logarithm of the ratio is log P. Here the partition coefficient measures how hydrophilic or water loving or hydrophobic the water fearing the chemical substance is. So if there are some organic solvents like ethyl acetate, or those have the lower density than water, they will be on the upper layer and water will be the lower layer. And the solvents which has a higher density than water like dichloromethane, so they will be in the lower layer and water will be on the upper layer. Okay, so that's how the separation happens between water and the other solvent. So the hydrophobic compounds, they have the high value of P. P is basically to measure how the compound is hydrophobic. Greater the value of P is the greater the particular compound is, the hydrophobicity of the compound is greater. Okay. So this was the problem number 3. Now I will go to the next problem. Okay, there is a mistake in the numbering. Okay, so it will be problem number 4. So problem number 4 says the correct equation to find out the substituent hydrophobicity constant pi x equal to what? Your options are number A log px minus log pH, number B log px by log pH, number C log px plus log pH, and number D log px into log pH. What is the correct equation to find out the substituent hydrophobicity constant pi x? This is our problem number 4. There is a mistake. I will just rectify it later. So what is the correct equation to find out the substituent hydrophobicity constant pi x? What is the correct equation to find out the substituent hydrophobicity constant pi x? Log px minus log pH. Number B log px by log pH. Number C log px plus log pH. Number D log px into log pH. Let me check the chat box. Okay, A is the given answer. Let me check if A is the correct answer or not. So the correct answer will be, yes, it is number A, log px minus log ph. Okay. So just let me first rectify the numbering. So the substituent hydrophobicity constant is denoted by the pi. Okay, so the pi x here is denotes any substituent, like for example chloro, nitro, amine. X is that particular substituent. So substituent hydrophobicity constant pi. So we have seen how hydrophobicity of a compound can be quantified using the partition coefficient p that we have discussed in our last slide, right? So in order to get the p, we have to measure it experimentally, and that means that we have to synthesize the compounds. It would be much better if we could calculate the p theoretically and decide in advance whether the compound is worth synthesizing. QSR would then allow us to target the most promising looking structures. For example, if we were planning to synthesize a range of the barbiturate structures, we could calculate the log p values for them all and concentrate on the structures which have the log p values closest to the optimal log p0 value of the barbiturate. Partition coefficients can be calculated by knowing the contribution that various substituents make to the hydrophobicity. This contribution is known as the substituent hydrophobicity constant, or pi, and is the measure of the how hydrophobic a constituent is relative to the hydrogen. The value can be obtained as follows. Partition coefficients are measured experimentally for the standard compounds such as benzene with or without the substituent X, and then substituent hydrophobicity constant pi x of the substituent X is the obtained for using following equation: pi x equal to log px minus log ph. Okay, where pH is the partition coefficient of the standard compound and px is the partition coefficient of the standard compound with the substituent X. The positive value of pi indicates that the substituent is more hydrophobic than hydrogen, and a negative value indicates that the substituent is less hydrophobic. The pi values would range for substituent from known as that there is a table which I will mention here. So this is the values of the pi of the substituent. Okay, and these pi values are characteristic of the substituent that can be used to calculate how the partition coefficient of the drug would be affected if these substituents were present. The p value of the lead compound would have been measured experimentally, but once that is known, the p value of the analogs can be calculated simply. So this is the range of the substituents. So pi aliphatic substituents. So when these functional groups are attached to aliphatic structure, the pi values are like this. For CH2 is 0.50, for tertiary butyl group it is 1.68, for hydroxyl group it is minus 1.416. So the positive value indicates the compounds are more hydrophobic than hydrogen. Negative value indicates the compounds are less hydrophobic than hydrogen. Okay. OMe is the 0.47, CF3 is 1.07, chlorine is the 0.39, bromine is the 0.60, and fluorine is the minus 0.17. So all these are for aliphatic substituent, and for aromatic substituent the values for methyl is 0.52, for tertiary butyl 1.68, for hydroxyl it is 0.67, for OMe it is minus 0.02, for CF3 1.16, for chlorine 1.71, for bromine 0.86, and for fluorine it is 0.14. Okay, so all these are calculated values. Okay, now these are experimental values which have been calculated. So this table you may remember or you may not be remember. Sometimes mathematical questions can be given from this type of things, and so the values will be given for some cases, or some cases you might have to calculate the values itself. Okay, we will see it in our next question because I have a related mathematical equation in our next question. So let us go to the next problem. So it will be problem number 5. It says that given the following log P values, what is the log P value of the metachlorobenzamide? Here you have the for the normal benzene structure, the log P value is 2.13. For chlorobenzene, log P value is 2.48. For benzamide, the log P value is 0.68. Right? And you have to calculate for the metachlorobenzamide. Your options are number A 1.97, number B 2.01, number C 1.35, number D 2.29. The value of log P value of benzene is given, log P value of chlorobenzene and benzamides are also given. You have to calculate the log P value of the metachlorobenzamide. I will give you some time. Try to solve the question. It is very easy. I have already discussed in the last slide how to do this type of calculations, and I know all of you can do it. Try to solve the question. I will wait for a minute, then I will show you how to do it, okay? Let me check the chat box. Okay, I got one answer which is saying it is the number B. Let me check if B is the correct answer or not. So the correct answer will be, it is the number C, 1.35. Okay, let me now show you how to solve these questions. The table we have earlier seen in the last slide, it is not possible all the time to remember or to memorize all the values. So how to calculate these things if we do not remember the values, okay? So we have to find out the log P of the metachlorobenzamide, right? So this metachlorobenzamide, the log P value will be equal to log P value of benzene which do not have any substituent, and then we have to just add the substituent constant or pi values of the substituents, okay? Like the pi for the chloro and pi for the CONH2 group. So this is we have to calculate. So now this pi chloro value, okay, is what will be the value? The value is the value of the chlorobenzene, and then we have to subtract the value of the benzene. Then we will get the value of the pi value of the chloro. Okay, so the pi value, the pi chloro value will be the log P value of the chlorobenzene minus the log P value of the benzene, which give us the pi value of the chloride, which is 2.84 minus 2.13 equal to 0.71. For CNH2, CONH2 it is the same. For benzamide the log P value is 0.64. We have to subtract the benzene log P value, which is 2.19, and the substituent value of this pi value of the CONH2 is the minus 1.49. So now the log P value of the metachlorobenzamide will be 2.13, which is the value of the benzene. We have to add the value of chloride, so 0.71, and also minus 1.49, which is 1.35. Okay, so this is the correct answer, and this is how you have to solve this equation. You can also take the help of the table if the table is given, because all the time it is not possible to memorize the values, but this is a very simple way how you can by which you can just calculate the particular thing is this particular problem easily. Okay, so this was the correct answer, number C. Now I will go to the next problem. So it says that which of the following statements regarding the drug salmazole is false? Your options are: number A, the drug is free from the CNS side effects. Number B, the log P value of the drug is 1.17. Number C, the 4-OMe group was replaced by 4-SOMe group. And number D, the log P value of the drug is 2.59. Which of the following will statement regarding the drug salmazole is false? Number A, the drug is free from the CNS side effects. Number B, the log P value of the drug is 1.17. Number C, the 4-OMe group was replaced by the 4-SOMe group. Number D, the log P value of the drug is 2.59. Let me know in the chat box A, B, C or D which you think is the correct answer. Let me check the chat box. Okay, I think there is no answer for this question. Okay, let me check what is the correct answer and let us discuss it a bit. So the correct answer for this problem will be number D. The log P value of the drug is 2.59 and there is an error in the typography. I will just correct it. So this is our problem number 6. Okay. So number D, option number D, which is the log P value of the drug is 2.59, it is incorrect. So alternative, the log P value to remove the CNS side effects. So the cardiotonic agents I, okay, so this was found. So I means, so this is the basic structure. Okay, and in this basic structure we have, so this is the imidazopyridine structure, and here we have a side linkage where is the R, so R equal to OMe in case of the structure, and number 2 is R equal to SOMe, which is the salmazole. When R equal to SOMe, the drug is known as the salmazole drug. So the cardiotonic agent 1 was found to produce the bright visions in some patients, which implied that it was entering the CNS or central nervous system. This was supported by the fact that the log P value of the drug was 2.59. In order to prevent the drug entering the CNS, the 4-OMe group was replaced by 4-OSSOMe group. This particular group is approximately of the same size of the methoxy group but more hydrophilic. The log P value of the new drug salmazole is found to be 1.17. The drug was now too hydrophilic to enter the CNS and was free from the CNS side effects. So this is a very good example how the log P value, altering the log P value of the particular drug will alter its side effects, which can actually contribute to for removing its side effects. Okay, so salmazole is a very good, example of this phenomena. So statement number D, which says that the log P value of the drug is 2.5 nm, it is false. The correct log P value of the drug is 1.17, which is more hydrophilic than the previous one, and which removes the CNS toxicity from this property, drug property. Okay. So this was our problem number 6. Now I will go to the next problem. So this is the problem number 7. So which of the following statements regarding the standard deviation is true? Your options are: number A, standard deviation is a measure that is used to quantify the variation or the dispersion of the data set. Number B, a low standard deviation indicates that the data points are spread over a wide range of values. Number C, a high standard deviation indicates that the data points are close to mean data of the data mean of the data set. And number D is none of the above. Which of the following statements regarding standard deviation is true? Again, I will repeat. Your options are: number A, standard deviation is a measure that is used to quantify the variation or dispersion of the data set. Number B, a low standard deviation indicates the data points are spread over a wide range of values. Number C, a high standard deviation indicates the data points are close to the mean of the data set. And number D, none of the above. Which of the following statements regarding the standard deviation is true? I will give you a minute, then I will check the chat box. Read the options carefully and then let me know A, B, C or D, which of the following you think is the correct answer. Let me check the chat box. Okay, one answer I have got, number A. Let me check if number A is the correct answer or not. So the correct answer for this problem will be yes, number A. Standard deviation is the measure that is used to quantify the variation or dispersion of a data set. So basically the standard deviation is D or S is the measure that is used to quantify the amount of variation and dispersion of the dataset values. Okay, so the important thing is like this: the low standard deviation value. This indicates the data points tend to be close to the mean, also called the expected value of the set, while a high standard deviation indicates that the data points are spread out over a wide range of the values. Okay, so this is important. What this low standard deviation and high standard deviation they denotes. So low standard deviation indicates that the data points tend to be close to the mean, also called the expected value of the set, whereas a high standard deviation indicates that the data points are spread over a wide range of the values. Other statistical tests are also conducted to validate this trend. So this is the calculation, the some equation which is used to calculate the standard deviation Sx equal to root this summation of from I equal to 1 to n, where this n is the n number of points. Okay, the xi minus x mean x bar the whole square divided by n minus 1, where n is the number of the data points, xi equal to the each values of the data, and x bar equal to mean of the xi. Okay, so this is the example how the normal distribution curve, it is known as the normal distribution curve, it looks like. So standard deviation, the statistic that measures the dispersion of a data set relative to its mean and is calculated as the square root of the variance. Okay, so statement number A, it was the true regarding the standard deviation. So problem number 7. Okay, now I will go to the next problem. Yeah. So next problem, it says that which of the following statements regarding the Craig's plot is false? Your options are: number A, it is a plot where x-axis indicates the value of the pi factor and the y-axis indicates the value of the sigma factor. Number B, this plot shows the close relationship between pi and sigma. Number C, from this plot it is impossible to tell which substituent have the positive pi and the sigma values and which substituent have a negative one. Number D, from this plot the substituents have the similar pi values and the similar sigma values can be observed. Again, I will repeat, which of the following statements regarding the Craig's plot is false? Your options are: number A, it is a plot where x-axis indicates the value of the pi factor and y-axis indicates the value of the sigma factor. Number B, this plot shows the close relationship between pi and sigma. Number C, from this plot it is possible to tell which substituent have the positive pi and sigma values and which of the substituent have the negative one. Number D, from this plot the substituents having the similar pi values and the similar sigma values can be observed. Which of the following statements is false? Read the options very carefully, think, and then let me know in the chat box which of the following you think is the correct answer for this problem. I will wait for a minute, then I will check the chat box and I will discuss it, okay? Let me check the chat box. Okay. I have not received any answer for this question. It's fine. We will just discuss it. So, the statement which is false regarding the Craig's plot will be number B. This plot shows a close relationship between the pi and sigma. This statement is false. So, although the tables of pi and sigma factors are readily available for a large range of substituent, it is often easier to visualize the relative property of the different substituent by considering a plot where the y-axis is the value of the sigma factor and x-axis is the value of the pi factor. Such a plot is known as the Craig plot. The example shown here, this is the Craig plot, okay, comparing the value of the pi and sigma for the various substituent. So the Craig plot is a pi and a sigma factor for the para-aromatic substituent. There are several advantages to use of the such Craig plot. The plot shows clearly that there is no overall relationship between the pi and sigma. Okay, the various substituents are scattered all around the four quadrants of the plot. It is possible to tell at a glance which substituent have the positive pi and the sigma parameters, which substituent have the negative pi and sigma parameter, and which substituent have one positive and one negative parameter. It is easy to see which substituent have the similar pi value. For example, the ethyl, bromo, trifluoromethyl, and trifluoromethyl sulfonyl groups are all approximately on the same vertical line on the plot. In theory, these groups could be interchangeable on the drug, whereas the principal factor affecting the biological activity is the pi factor. Similarly, groups which form a horizontal line can be identified being the isoelectronic or having similar sigma values like COOH, chloro, bromo, and iodo. Okay, so this is your Craig plots and comparing the values. Okay, so number B, which says that this plot shows a close relationship between this both of them. So it is false because it's clearly written here that the plot shows clearly that there is no overall relationship between the pi and sigma. The various substituents are scattered all around the four quadrants of the plot. Okay, so there is no not any kind of relationship between these two factors. So now we will go to the next problem. So problem number 9, it says that what term is used to describe the process by which the best line is fitted through a set of data points on the graph. Again, I will repeat, what term is used to describe the process by which the best line is fitted through a set of the data points on a graph. Your options are number A, linear regression analysis by the least square method. Number B, linear fitting analysis by the least square method. Number C, linear fitting assessment by the least square method. Number D, linear regression assessment by the least square method. What term is used to describe the process by which the best line is fitted through the set of the data points on the graph? Your options are number A, linear regression analysis by the least square method. Number B, linear fitting analysis by the least square method. Number C, linear fitting assessment by the least square method. Number D, linear regression assessment by the least square method. A, B, C or D, let me know which of the following will be the correct answer for problem number 9. I will wait for a minute or two, then I will check the chat box. What term is used to describe the process by which the best line is fitted through a set of data points on the graph? Let me check the chat box now. Okay, there is no answer till now. It's fine. Let me just show you the correct answer. So the correct answer for problem number 9 will be number A, linear regression analysis by the least square method. So what is the least square method? So it is a form of mathematical regression analysis used to determine the line of the best fit for a set of data, providing the visual demonstration of the relationship between the data points. So linear regression analysis or least square method is one technique for estimating a line best fit. Once the equation for the best line of the best fit has been determined, forecast can be made. The least square method or linear regression analysis involves using the following formula for A and B, where y equal to a plus bx, where b equal to n summation of xy minus sum of x sum of y divided by n sum of x square minus sum of x sum of x whole square. So where an a equal to y bar minus b x bar, where n equal to number of the data pairs and the number of the data, x bar equal to the mean of x values of the all points of data, and y bar is the mean y value for all pairs of the data. Okay, so this is the formula of linear regression and all about it. So problem number 10, it says that the value of the sigma m for the nitro substituent is 0.71, whereas the value of the sigma p is the minus 0.78. What does this sigma m and sigma p stand for? Your options are number A, the electronic effect of the nitro substituent at the metapara positions respectively. Number B, the hydrophobic effect of the nitro substituent at the metapara positions respectively. Number C, the steric effect of the nitro substituent at the metapara position respectively. Number D, the acidic effect of the nitro substituent at the meta and para position respectively. What does this pi m and sigma m and sigma p stands for? Electronic effect, hydrophobic effect, steric effect of or acidic effect at the meta and para positions respectively. Let me know. A, B, C or D, which of the following you think is the correct answer? Let me check the chat box. Okay, it's fine. So for problem number 10, the correct answer will be this sigma M and sigma P stand for the electronic effect of the nitro substituent at the meta and para positions respectively. So option number A. So the electronic effects of the various substituent will clearly have an effect on the drug's ionization or polarity. This in turn may have an effect on how easily a drug can pass through the cell membranes or how strongly it can interact with the binding site. It is therefore useful to measure the electronic effect of the substituent. As far as the substituents on an aromatic ring can concern, the measure is used to known as the Hammett substituent constant or sigma. This is the measure of the electron withdrawing or electron donating ability of the substituent and has been determined by the measuring the dissociation of the series of the substituent benzoic acid compared with the dissociation of the benzoic acid itself. The benzoic acid is the weak acid and only partially ionizes in water. An equilibrium is set up between the ionized and non-ionized form where the relative proportion of this species is known as the equilibrium or dissociation constant Kh. The subscript H signifies there is no substituent on the aromatic ring. Okay, so Kh equals to PHCO, so ionized form of benzoic acid, the concentration divided by the concentration of benzoic acid. So when the substituent is present in the aromatic ring, the equilibrium is affected. The electron withdrawing groups such as the nitro group result in the aromatic ring having a stronger electron withdrawing and stabilizing influence on the carboxylate ion, and so the equilibrium will shift more to the ionized form. Therefore, the substituent benzoic acid is the stronger acid that has a large Kx value, where X represents the substituent on the aromatic ring. If the substituent X is an electron donating group, such as an alkyl group, then the aromatic ring is less able to stabilize the carboxylate ion. The equilibrium shifts to the left, calculating with a weaker acid with a smaller Kx value. The Hammett substituent constant sigma x for the particular substituent x is defined as the following equation: sigma x equal to log kx by kh equal to log kx minus log kh. Okay, the benzoic acids containing electron withdrawing substituent will have a larger kx values than the benzoic acid itself, and therefore the value of sigma is of the electron withdrawing substituent will be positive, and substituents such as the chlorocyano CF3 will have positive sigma value. Benzoic acids containing electron donating substituent will have a smaller Kx value than the benzoic acid itself, and hence the value of sigma x for an electron donating substituent will be negative. Substituents such as the methyl, ethyl or tertiary butyl will have the negative value of sigma. The Hammett substituent constant for the H is 0. The Hammett substituent constant takes into the account of both resonance and inductive effects. Therefore, the value of the sigma for a particular substituent will depend on whether the substituent is a meta or para. This is indicated by the subscript M or P on the sigma symbol. For example, nitro substituent have a sigma P of 0.78 and the sigma M of 0.71. In the meta position, electron withdrawing power is due to the inductive influence of the substituent, whereas at the para position, it is both play a role. So sigma P is the greater. So there will be this. Okay, so this sigma values denotes the electronic effect of the nitro substituent. Now I will go to the next problem. Problem number 11 says when two drugs act on the same receptor resulting in the drug interaction that is said to be which of the following? Your options are number A predictable, number B unpredictable, number C additive, number D empty therapy. When two drugs act on the same receptor and resulting the drug interaction is said to be not the which of the following: predictable, unpredictable, additive or empty therapy. Let me know A, B, C or D which of the following you think is the correct answer for problem number 11. When two drugs act on the same receptor, the resulting drug interaction is said to be which of the following: predictable, unpredictable, additive or empty therapy. Let me check the chat box. Okay, no answer is fine. I will just show you the correct answer. So, when two drugs act on the same receptor, resulting drug interaction is said to be number C, additive. So this is known as the drug synergy. So when the drugs are given in combination, their effect can be antagonistic, subadditive, additive, or synergistic. So drug synergism arises when the therapeutic effect of two or more drugs are used in combination is greater than the sum of effects of the drugs administered individually. So one can think about the synergy by inhibiting the drug destroying enzyme. So we have already looked at this previously. For example, you can have an inhibitor of an enzyme that it destroys the drug, and this will result in synergy. This particular molecule may not have any therapeutic effects on its own, but it protects the drug from being destroyed. So as called the sensitizing of the drug and the sensitizing of the pathogen to the drug. But if the primary drug and inhibitors, the destroying enzymes are administered together, then together can have a synergistic effect. So we have already seen this in case of ampicillin and flavonoid. So they are administered together. So you can protect the ampicillin from the beta lactamases. So drug synergy, we have already looked at a part when we discussed a little bit earlier. So the drug synergy, when the drugs are given in combination, they can be antagonistic. That means that one drug going to cancel out the effect of the other, or it can be subadditive, means it's going to be little bit addition of in the efficacy, or it can be additive, which means the effect of A can be added to the effect of B and is going to be result in a combination therapy. But more importantly, when we look at the synergy, so here the synergy will refers to when the therapeutic effect of this two drugs can be used in combination is greater than the sum of the effects of the drug administered individually. So the difference between the additive and synergy is that that when I add these two drugs and concentration at which their efficacy is the net effect does not go up, but the synergy, the sum of the effects is larger than the individual drug. Okay, so this is about the additive effect and the drug synergy. Now let me go to the next problem. Problem number 12 says, which of the following resistant mechanism describes the function of the beta lactamase? Your options are number A, flux pump, number B, target mimicry, number C, drug inactivation, number D, target overproduction. Which of the following resistant mechanism describes the function of the beta lactamase? A flux pump, target mimicry, drug inactivation, and target overproduction. Let me know through the chat box A, B, C, or D which of the following you think is the correct answer for problem number 12. Which of the following resistant mechanism describes the function of beta lactamase? Let me check if there is any answer in the chat box. Okay, that's fine. So, mechanism of resistance is the drug inactivation. In case of the beta-lactamase, the drug resistance arises from the drug inactivation. So, there are the type of resistance called the drug inactivation. Some bacteria can use different methods to render the antibiotic useless, especially by destroying the antibiotic structure. Okay, so in case of the beta-lactamase, what we know, this particular this beta-lactam ring is very crucial for the activity of the beta-lactamase, right? So sometimes, so what is the so the drug resistance happens when this particular beta-lactam ring is destroyed by the ring is destroyed. Okay, so this is the mechanism of action how beta-lactamase works. So we have the two type of, one is the serine beta-lactamase and then second one is the metallo-beta-lactamase. So in case of the serine beta-lactamase, so this is on the cell wall of the bacteria. We know there is a glutamine residue, lysine residue, and serine residue. The serine residue acts as a nucleophile and attack on the beta-lactam ring, and then the water. This is the first step is the acylation step, and the second step is the deacylation step. When the incoming the water, there is a water in the vicinity, and this water attacks on this carbonyl carbon, and we have our this normal, this beta-lactam is here, beta-lactam is here, and there is have this which is in beta-lactamase. Okay, so that's how this beta-lactamase acts on the beta-lactam ring on the penicillin antibiotics. Okay, and this is that for the metallo-beta-lactamase. So some beta-lactamase have this metallo zinc core with for 3 histidine residues and 1 hydroxyl residue. Here this hydroxyl residue acts as the nucleophile and attacks on the beta-lactam ring of the penicillin and breaks this ring, and we all know this beta-lactam ring of penicillin is very crucial, and that is how the beta-lactamase destroys this beta-lactam ring and then hinders this activity of this our penicillin antibiotics. Okay, so this type of resistance of the drug inactivation is known as a drug inactivation. Now I will go to the next problem. Problem number 13 says the role of the efflux pump in the drug resistance is due to number A, metabolizing the drug before reaching the active site. Number B, inactivation of the drug. Number C, destroying the drug concentration inside cell. Number D, none of the above. What is the role of the efflux pumps in the drug resistance? Number A, metabolizing the drug before reaching the active site. Number B, inactivation of the drug. Number C, decreasing the drug concentration inside the cell. Number D, none of the above. Let me know A, B, C or D, which of the following you think is the correct answer, or which of the following you think is the correct role of the efflux pump in the drug resistance. So what is the role of the efflux pumps in the drug resistance? One time I will check the chat box. Okay, let us discuss it. So the role of the efflux pumps in the drug resistance is due to decreasing the drug concentration inside the cell. So alter distribution of the drug distribution to the site of the action. Efflux pumps are transmembrane proteins that are often highly expressed by the microorganisms and the tumor cells that act as the transporters by binding to the drugs and carrying them to the out of the cell before they can exert then a therapeutic effect. Some transporters known as the multi-drug resistance pump are quite broad in their specificity and can efflux a variety of natural and the synthetic drugs. Both specific and broad specificity efflux transporter systems have been identified that contribute to the bacterial resistance to a broad range of the antibiotic drugs. Okay. Efflux pumps have the clinical relevance for both bacteria and eukaryotes. The efflux pumps can confer MDR resistance and they can add themselves to other resistance mechanisms. Efflux pump can be seen as a new target. Efflux pump inhibitor by blocking the pumps will restore the drug's activity. Okay, so basically antibiotic. So this is a one diagram which indicates this efflux pump. So this is the antibiotic resistance bacteria. We have this pumps. This is the green ones are known as the efflux pump. So sometimes the antibiotics fails to penetrate the membrane, and sometimes when inside the membrane, the efflux pump they actually pulls out the drugs out inside the cell. So this is the efflux pump, and the efflux pump actually recently in recent years it had become a very good target for the antibacterial drug discovery, because efflux pump inhibitors are very crucial inhibitors which blocks this efflux pump, and by blocking it will restore the antibacterial activity. So in case of multi-drug resistant cases, this efflux pump inhibitor they act as a very good new drug target. Okay. So the role of the efflux pump in drug resistance is due to decreasing the drug concentration inside the cell. Now I will go to the next problem. Problem number 14 says drug resistance of trimethoprim is generated due to dash. Your options are number A, efflux pump, number B, sequential blocking, number C, mutation in the target, number D, insufficient drug concentration. Drug resistance of trimethoprim is generated due to which of the following is the correct reason? Efflux pump, sequential blocking, mutation in target, or insufficient drug concentration. What will be the correct answer? What will be the correct reason of drug resistance of trimethoprim? Efflux pump, sequential blocking, mutation in target, or insufficient drug concentration. So drug resistance of trimethoprim is generated due to which of the following will be the correct answer? Let me check it once. Okay, let us discuss it. So drug resistance of trimethoprim is generated due to number C, mutation in target. So let us say you have a protein. So the mutation can occur here, and this results in the conformational change which changes the binding pocket of the enzyme. So this is now there's not very good binding, good at binding anymore. For example, resistance to the antibiotic trimethoprim derives from a single mutation of the single amino acid of the target enzyme, which is the dihydrofolate reductase. The properties of the singly mutated an enzyme differ somewhat than the normal enzyme, but it still binds to the substrate dihydrofolate. Okay, so dihydrofolate reductase is the enzyme where the trimethoprim acts as the inhibitor, but a single point mutation changes the conformation such that now the antibiotic trimethoprim is unable to bind on this particular enzyme, but the enzyme still binds with the substrate. Okay, so this is the reason of the drug resistance of trimethoprim. So this is the complete this the dimethoprim how is the way by which it happens. Okay, so this folic acid metabolism, the first is the pteridine and paraminobenzoic acid which makes the dihydrofolate acid, and the enzyme is dihydrofolate synthesis which is inhibited by the sulfonamides. Then the dihydrofolate acid with in presence of glutamate it converts to the dihydrofolic acid, and the enzyme is dihydrofolate synthesis, and dihydrofolic acid converts to tetrahydrofolic acid, and the enzyme is the dihydrofolate reductase, and trimethoprim inhibits this particular enzyme. Okay, but the single point mutation in this particular enzyme makes this trimethoprim now unable to bind to this particular enzyme and inhibit it. So the reason of the drug resistance of trimethoprim is due to the mutation of the target. Okay, now let me go to the next problem. So problem number 15 says, which of the following is not a mechanism of resistance of the quinolones? Your options are number A, lack of potential gradient, number B, single point mutation, number C, active efflux system, number D, mutation in the genes of the DNA guides. Which of the following is not a mechanism of resistance of quinolones? Again, I will repeat: number A, lack of the potential gradient, number B, single point mutation, number C, active efflux system, number D, mutation in the genes of the DNA guides. Let me know A, B, C or D, which of the following is not a mechanism of resistance for quinolones? Which of the following is not a mechanism of resistance of quinolones? Okay, I got one answer which is saying it's number B. Let me check if number B is the correct answer or not. So, for problem number 15, the correct answer is number A, lack of potential gradient. It is not a mechanism of resistance for the quinolone. So basically bacterial resistance to the quinolone, the resistance can emerge during the therapy, especially with the S. aureus and the P. aeruginosa. The single mutation is sufficient to cause the resistance. Resistance and chromosomal rather than the plasmid mediated mutation can occur in the genes for the DNA guidance, the topoisomerase type 2, and appear to be the primary site of the S pneumonia and other gram positives, active efflux pump present in both gram positive gram negative bacteria. So these first three, one is the single point where single mutation. Okay, the single point mutation, then mutation in the DNA guidance or topoisomerase type 2 and active efflux pump. All these are the method of the drug resistance for the quinolone, not the lack of the potential gradient. Okay, so the correct answer will be number A, because the lack of potential gradient is not a mechanism of resistance of the quinolone. Okay. Now I will go to the next problem. Problem number 16 says aminoglycoside resistance is due to, your options are number A, target mimicry, number B, target overproduction, number C, efflux pump, and number D, lack of the electrical potential gradient across the bacterial membrane. Aminoglycosides resistance is due to target mimicry, target overproduction, efflux pump, or lack of the electrical potential gradient across the bacterial membrane. A, B, C, or D. Let me know which of the following you think is the correct answer for problem number 16. What is the correct reason for the aminoglycosides resistance? So aminoglycoside resistance is due to, what will be the correct reason? Okay, let me show you the correct answer and let us discuss it a little bit. So the resistance of the aminoglycosides is basically due to Number D, lack of an electrical potential gradient across the bacterial membrane. Let me discuss it a little bit. So, aminoglycoside antibiotic resistance can arise from the lack of the quinones that mediate the drug transport, or from the lack of an electrical potential gradient required to drive the drug across the bacterial membrane. Aminoglycoside is a medicinal and bacteriologic category of traditional gram-negative antibacterial therapeutic agents that inhibit the protein synthesis and contain as a portion of the molecule an amino modified glycoside or sugar. Aminoglycoside antibiotics display the bactericidal activity against the gram-negative aerobes and some anaerobic bacilli, which resistance has not yet been arised, but generally not against the gram-positive and anaerobic gram-negative bacteria. So one of the ways which can occur in which this can occur is by the changing the net charge on the membrane. So if you can, for example, if there is a glycerol, the membrane can be changed to lysyl phosphorylglycerol group. Then it converts anionic group to the cationic group. So the change can affect the permeability of the drug. So if the permeability of the drug goes down, clearly its efficacy will go down. In this way, the drug with some charge can actually now be repelled. So aminoglycoside antibiotic, for example, become inactive because the quinones that mediate the drug transport, or from lack of the quinones that mediate the drug transport, or from the lack of an electrical potential gradient required to drive the drug across the bacterial membrane. So these two can result in the decreased uptake of the aminoglycosides, which is going to result in the resistance. Okay. So this is the reason of the aminoglycoside resistance, that is the lack of the electrical potential gradient across the bacterial membrane. Now I will go to the next problem. Problem number 17 says, which of the following is the correct reason of the drug resistance of kanamycin? Your options are number A, elimination of the hydroxyl group, number B, phosphorylation of the hydroxyl group, number C, efflux pump, number D, target overproduction. Which of the following is the correct reason for the drug resistance of the kanamycin? Elimination of the hydroxyl group, phosphorylation of the hydroxyl group, efflux pump, and the target overproduction. Let me know A, B, C or D which you think is the correct reasons for the drug resistance of the kanamycin. Let me check the chat box. Okay, let me share the correct answer for this problem. So the correct reason of the drug resistance of the kanamycin is the number B, phosphorylation of the hydroxyl group. So this is the structure of the kanamycin, and as you can see, has this sugar structure, the sugar backbone, which has a couple of hydroxyl group, and the phosphorylation of this hydroxyl group will render this particular kanamycin inactive, and this is the reason of the drug resistance. Okay, so another potential approach to countering the resistance is to modify the antibiotics such they can gain resistant to the mechanism of resistance used against them. For example, kanamycin is an aminoglycoside which has no longer can be used because the reason can because the resistant bacteria can phosphorylate one of the hydroxyl group presents, and active analog has been synthesized, okay, which replaces the susceptible alcohol with a ketone. This ketone is in equilibrium with the hydrated GM diol. When phosphorylation occurs on the diol, the phosphate group thus acts as a good leaving group, and the ketone is regenerated. In vitro tests show that this agent was active against the strains of bacteria which are resistant to the kanamycin. So this is the ketone, which is the analog of the kanamycin, and this is the hydrated diol. Okay. And this is the phosphorylation. Now, if the phosphorylation occurs, it has an equilibrium which happens. So this is the analog of kanamycin, which is resistant to phosphorylation, and this is the and the phosphorylation, which was the main reason for the drug resistance of the kanamycin. Okay, so the correct answer is number B, which number B. Now I will go to the next problem, problem number 18, which says that which of the following mechanism is followed for the resistance of the sulfa-based antibiotics. Your options are number A, target mimicry, number B, efflux pump, number C, drug inactivation, number D, target overproduction. Which of the following mechanisms is followed for the resistance of the sulfa-based antibiotics? Target mimicry, efflux pump, drug inactivation, or the target overproduction? Which of the following mechanisms is followed for the resistance of the sulfa-based antibiotics? Let me know A, B, C, or D. Which of the following you think is the correct answer? Which you think is the correct reason or the correct mechanism for the resistance of the sulfa-based antibiotics? Target mimicry, efflux pump, drug inactivation, or target overproduction? Which of the following mechanisms is followed for the resistance of the sulfa-based antibiotics? Which of the following mechanisms is followed for the resistance of the sulfa-based antibiotics? Target mimicry, efflux pump, drug inactivation, target overproduction. Let me check the chat box now. Okay, let us discuss it. So, the mechanism which is followed for the resistance of the sulfur-based antibiotics will be number D, target overproduction. So overproduction of the substrate or the ligand for the target protein, overproduction of the substrate or for the target enzyme would competitively block the ability of the drug to bind at the active site. For example, overproduction of the para-aminobenzoic acid as the mechanism of resistance for the antibacterial sulfa drugs. So we all know, I have already discussed it for in some of my classes where we were discussing about the sulfur drugs, that sulfonamides, they are the competitive inhibitors for this particular enzyme, right? So the competitive enzymes, what does they do? They compete with the substrate for that particular enzyme and inhibit and bind to the particular active site of the where the substrate binds, and by mimicking the structure of the substrate. So for this particular enzyme, which is the dihydropteroate synthetase, the normal substrate is the para-aminobenzoic acid. And if you can see the structure of the para-aminobenzoic acid, the interactions which it forms with the enzyme is very similar to the sulfonamides, and that is why the sulfonamides they compete with the this para-aminobenzoic acid and competitively inhibits and binds to the particular substrate. But in cases where the para-aminobenzoic acid, which is the normal substrate, which is overproduced, so what it will do, the overproduction of this particular substrate, it will remove the sulfonamides from the active site and occupy the active site of the enzyme. Okay, so that is the sulfonamides. So that is the mechanism of the drug resistance for the sulfur-based antibiotics, and this is the same. It is mostly same for all the competitive inhibitor drugs. With the drugs which acts as a competitive inhibitor, in all the cases, this is same. This target overproduction, it causes the resistance for this antibiotics. Okay. So number D was the correct answer. Now problem number 19, it says that which of the following mechanisms is the major reason for the MDR-TB, which is the multi-drug resistance tuberculosis. Your options are number A, target mimicry, number B, target overproduction, number C, mutation, and number D, efflux pump. Which of the following mechanisms is the major reason for the MDR-TB, multi-drug resistance tuberculosis? Target mimicry, target overproduction, mutation, or the efflux pump? Which of the following mechanisms is the major reason for the MDR-TB? Let me know. Which of the following mechanisms is the major reasons for the multi-drug resistant tuberculosis? Let me check the chat box once. Okay, let me discuss it. So in case of multi-drug resistant tuberculosis, the mechanism of the major reason or the mechanism of drug resistance is mutation, which is the number C. So drug resistance by mutation, bacteria multiply at such a rapid rate that there is always a chance that a mutation will render a bacterial cell resistance to a particular agent. This feature has been known for a long time and is the reason why patients should complete a full course of antibacterial treatment even though their symptoms may have disappeared well before the end of the course. If this is the rule, if this rule is adhered to, the vast majority of invading bacterial cell will be wiped out, leaving the body's own defense to mop up any isolated survivor or resistance cells. If the treatment is stopped too soon, however, the body's defenses struggle to cope with the survivors. Any isolated resistant cell is then given a chance to multiply, resulting in a new infection, which will, of course, be completely resistant to the original drug. That this is a major factor in the appearance of the MDR-TB. So mutation can occur naturally and randomly and do not require the presence of the drug. Indeed, it is likely that the drug-resistant cell will present in the bacterial population even though the drug is encountered. This was demonstrated with the identification of the streptomycin-resistant cells from the old cultures of E. coli, which had been freeze-dried to prevent the multiplication before the introduction of the streptomycin to the medicine. Okay, so mutation is a major factor for the appearance of the MDR-TB. The last question of today's session, it says which term is used to describe the inhibition of two different enzymes in the same biosynthetic pathway? Your options are number A, parallel inhibition, number B, sequential blocking, number C, empty therapy, number D, enzyme inhibition. Which term is used to describe inhibition of two different enzymes in the same biosynthetic pathway? Parallel inhibition, sequential blocking, empty therapy, or enzyme inhibition? Which term is used to describe inhibition of two different enzymes in the same biosynthetic pathway? Let me know A, B, C, or D, which you think is the correct answer. Let me check the chat box. Okay, number B is the given answer. Let me check if number B is the correct answer or not. So, the inhibition of two different enzymes in the same biosynthetic pathway is known as, yes, it is number B, the sequential blocking. So the inhibition of two or more consecutive steps in the metabolic pathway is known as a sequential blocking. The reason it is effective is because it is difficult, particularly with the reversible inhibitor, to inhibit an enzyme 100%. If less than 100% of this enzyme activity is blocked, the metabolic pathway has not till shut down. Okay. So sequential blocking, for example, is a pathway A to B to C to D to E. If we block one pathway, like for example B to C, but if this we try with the let's say it is an enzyme and inhibition, and if it is not 100%, then the metabolic pathway is all still on. But when combined use of inhibitor, two consecutive enzymes in the pathway, it is possible to block the metabolic pathway completely. So if let's say if this is the pathway, if we block it two times in two different steps, so it may altogether it is possible to complete the for completely shut down this particular metabolic pathway. So let us say A goes to B, goes to C, goes to D, goes to E. Now, so this is the metabolic pathway and we want to inhibit. So let us say we develop an inhibitor for this step. Okay. So here, because D does not go converted to C, the inhibition you do not get formation of the E. But less than 100% of this occur, because we are looking at the reversible inhibition, it is possible that you will never get a 100% inhibition. So if you less than 100% inhibition of the enzyme activity is blocked, then the metabolic pathway has really not been truly shut down. There is always going to be the some amount of E that is produced. But when you use inhibitors of two consecutive enzymes in the pathway, so what we will go, A is going to B is going to C. What is going to be happen? We will not just block this, but perhaps we are also block this. Okay, so here we will develop two different inhibitors. So this is the which can work in synergy. So this is known as the technique of the sequential blocking. So this was the last question of today's session. So today we discussed majorly regarding the QSAR, various parameters of QSAR and various tactics, and we discussed about the various pathways through which the drug resistance can happen. We discussed about the drug inactivation, efflux pump, a little bit about of the potential gradient and the other mutation and so on. We discussed about the synergy and also we discussed about the sequential blocking. So this was all for today. This is our week 12 live session. So this is our last Friday class, our last live session for the regular live sessions, and the last class which we will have is on 13th of April, which is on the day next tomorrow, means on Sunday, and the class will be from 11 a.m. So this is my email ID. If you have any doubt or any query, or you want me to let you discuss any topic of your interest which you didn't understood did not understand before earlier, so let me know by email so that I can include it in the our summary class. Okay, so this is my email ID. If you have any topic which you want me to discuss it further, you can just let me know through the email. This class presentation and the recording I will be sharing to you, so you can check it from there. If you have any problem, any query, please let me know. So thank you all for joining. I will close the session for today. Feel free to let me know by the email if you have any doubt or any query. So I am closing the session for today. Thank you.